The idea that gut bacteria can influence hunger is no longer fringe. It's rapidly becoming central to how scientists understand appetite regulation. But an even more provocative question is emerging. Can microbes actually turn off hunger? New findings suggest the answer may be yes, and the mechanism involves bacterial components like flagellin, which appear capable of engaging gut receptors and triggering neural feedback loops that reduce food intake. This shifts our understanding of satiety from a purely host-controlled process to a shared dialogue between human biology and the microscopic organisms that live within us. Flagellin is a protein that forms the tail-like structures, or flagella, of many motile bacteria. While it might seem like a structural feature with little metabolic consequence, the human gut has evolved receptors that detect flagellin as a kind of microbial fingerprint. These receptors, particularly TLR5, toll-like receptor 5, are found in various parts of the gut lining and are capable of initiating immune responses. But what's now being discovered is that the activation of TLR5 doesn't just affect immune tone. It also feeds directly into gut-brain signaling circuits that govern feelings of fullness and the cessation of eating. When certain bacteria release flagellin, it binds to these gut receptors and sets off a cascade of effects. This includes the release of satiety hormones like peptide YY, PYY, and glucagon like peptide 1 GLP-1, which act both locally on the gut and systemically via the bloodstream to signal to the brain that the body no longer needs food. But the effect isn't just hormonal. It's also neurological. Evidence shows that gut receptors activated by bacterial products can engage neuropods, specialized cells that communicate with the vagus nerve. This creates a rapid, nerve-based feedback loop that transmits microbial information to the brain's appetite regulation centers, such as the hypothalamus, in real time. This mechanism suggests that microbial populations can fine-tune hunger not just by influencing energy extraction from food, but by sending specific stop signals to the brain. In animal studies, mice raised in sterile, germ-free conditions often exhibit abnormally high food intake and altered satiety signaling, which returns to normal after microbial colonization. In other experiments, introducing bacteria that release specific satiety-associated molecules, such as CLPB, a bacterial protein that mimics the structure of appetite-regulating hormones, has been shown to reduce food intake and body weight in animals. These findings imply that some gut microbes have evolved ways to manipulate host appetite for mutual benefit. If the host eats less but absorbs more efficiently, both the host and its microbial residents thrive. The implications are far-reaching. For individuals struggling with obesity or metabolic syndrome, microbial interventions could eventually offer a non-invasive and biologically harmonious strategy to reduce overeating. Instead of suppressing appetite through blunt pharmaceuticals, we might cultivate or engineer microbial strains that naturally modulate hunger through flagellin signaling or similar pathways. Probiotic or postbiotic therapies targeting TLR5 activation, or boosting the production of specific microbial proteins that engage satiety circuits, could help restore balance in dysregulated appetitive systems. But this also raises the possibility that microbial imbalance, or dysbiosis, may disrupt these hunger-regulating pathways. If beneficial flagellin-producing bacteria are depleted due to antibiotics, processed diets, or chronic inflammation, the gut's ability to sense satiety might become impaired. Without proper microbial cues, the body may interpret the absence of these signals as a sign to keep eating. This could explain why some individuals with metabolic disorders experience persistent hunger despite adequate caloric intake. Their gut-brain axis is starved of the microbial inputs it once relied on. There's also an evolutionary angle worth considering. Microbes that live in the gut have co-evolved with humans for millennia. From a symbiotic perspective, it's in their interest to avoid driving the host to overeat to the point of dysfunction. Satiety signals may thus reflect a cooperative equilibrium between species, one that favors stability, metabolic health, and sustainable nutrient sharing. But in modern environments, with ultra-processed foods, sedentary lifestyles, and frequent antibiotic use, this delicate microbial symphony is often thrown off balance. Still, the emerging science paints a hopeful picture. By better understanding which bacterial signals influence appetite, such as flagellin and other microbial metabolites, we may one day fine-tune hunger at its source, not by overriding brain chemistry, but by restoring the missing voices in the gut-brain conversation. Rather than fighting our biology, we would be coaching it back into alignment with the ecosystem that supports it. So, can microbes truly turn off hunger? Increasingly, the answer appears to be yes, not in an all-or-nothing way, but through a sophisticated, evolving partnership between bacterial signals and neural circuits. It's a reminder that appetite is not just a matter of willpower or metabolism. It is a dialogue, and the microbes have something important to say.